Hello and welcome. This is Karen Sharon, and I'd like to welcome you to this lecture on the essential elements of a vaccine trial protocol. The backbone of a vaccine trial is a well-crafted protocol, and GCP provides a guidance on the content and structure of a written protocol document, and I'm going to use this as a framework for the lecture. The first section, we're going to move through the background and trial design. What is a protocol? It's always good to start out with a definition, and the official definition of a protocol can be found under GCP 1.44. To remind you, throughout this course we'll be referencing sections of ICH GCP, and you can find these in your GCP textbook that you purchased for the course. If you didn't get the book, I highly recommend that you do get the book. You can also find ICH GCP guidelines online. They're a little bit more difficult to search through, but you can find them for free online. So, GCP defines the protocol as a detailed document that describes the objectives of the trial, the design, the methodology, including all procedures in the trial, the statistical considerations for the trial, and how the trial is organized, including the key players that will be involved in the trial. The protocol also provides details on the background, rationale, and real clear procedural instructions on how to conduct the trial, although some of this particular information may be found in supporting documents. The protocol document has a large audience. It's written by the sponsor and the investigator who designed the trial, and it serves as this guiding document for all those who are involved in the study. Vaccine trials are not a solitary endeavor, so if you're interested in getting involved in this business, realize that vaccine trials involve teams of dedicated individuals, and I've mentioned a few of the individuals here on this slide. Then, once a protocol is finalized or agreed upon, the protocol and the supporting documents are submitted for review to the National Regulatory Authority, which in the U.S. is the Food and Drug Administration, the FDA, and the Institutional Review Boards, or IRBs, that review the trial for ethical considerations. So back to GCP, 6.0 outlines the core elements of a clinical trial protocol, and they're shown on this slide. These elements are going to be the framework for this lecture. And the principles of GCP apply to all clinical trials. There's not specific GCP for vaccine trials, but in this lecture and in this course, I'm going to discuss them in the context of a vaccine trial. And to do this, I'm going to pull some examples from some of our trials at Johns Hopkins and trial information available on a public website, clinicaltrials.gov, and I have a reference to that on this slide. If you don't know about clinicaltrials.gov, I suggest you take a time to explore that site at another time. All trials that are conducted in some way through the U.S., either through review or with U.S. investigators, have to be registered on clinicaltrials.gov, and there's a lot of information out there you can find. Section 6.1 of GCP outlines the general information that should be included in a protocol. Now here's an example of that information using a trial I found on clinicaltrials.gov, and it starts with the title. There's an official detailed title that will give a lot of the specific technical names, and a summary or shorthand title. The purpose of the trial provided in this example was to evaluate an experimental vaccine to prevent diarrhea. The sponsors listed here and the collaborators and investigator. And in this particular example, you'll notice that principal investigator is Dr. Harrow, who's a colleague at the Center for Immunization Research here at Hopkins, and also one of the presenters in this course. So let's talk now about sponsoring the trial and monitoring. The protocol document needs to identify who is sponsoring the trial, and the sponsoring agency will be generally an organization or a company, could be a drug company, a biotech company, a government agency, a non-government agency, that's responsible for initiating, managing, and financing the trial. It has the overall umbrella responsibility for those tasks. Monitoring falls within those sponsor responsibilities. Now, the sponsoring agency or company may actually do the monitoring themselves, or they can delegate this task to another organization, which is called a contract research organization, or CRO, and there are many excellent CROs that serve in this capacity for many different trials, including vaccine trials. If the sponsor delegates tasks, those tasks in that arrangement needs to be identified in advance and needs to be documented on the protocol so everyone knows who's responsible for which elements of the protocol. Protocols may have something called a synopsis or a summary or an at-a-glance summary of key facts, and this slide lists some things you may find in a synopsis, the objectives and population, number of subjects. You can actually find a summary on clinicaltrials.gov for the trials that are registered there, so this is the kind of information that you would find. But we're going to move on to some more substantial sections of the protocol. So GCP 6.2 is the background section. And in order to propose an investigational vaccine trial, you have to provide information on the existing knowledge of the burden of disease the vaccine's targeted against and currently available therapeutic and/or preventive interventions that exist. It has to include information about the specific investigational vaccine that you're studying and the potential impact of the investigational vaccine on that disease burden. It will have a summary of all preclinical data, which means animal and laboratory data, as well as clinical trial data. That can be information from this vaccine if it's been tested before, or similar vaccines that are either licensed or who have been tested. The background would have a summary of known and potential risks and benefits of the product, and a justification for the routes of administration, the dose, and the schedule that's being proposed in the study. It would have a description of the population you're going to enroll, and then references to the published literature on the topic. It's a pretty substantial portion of the protocol. The next section is trial objectives. Every protocol has primary and secondary objectives, and the first two listed here on this slide are common. Safety is always one of the primary objectives. No matter what phase of a vaccine trial, you're evaluating safety. And vaccine trials evaluate the immune response of vaccinees as compared to those that receive the control of the placebo. So that's also one of the objectives. Then trials have specific objectives relating to that trial design, such as evaluating a dosage level or schedules or many different trial designs. Speaking of trial design, we're going to spend some time here discussing trial design. So GCP 6.4 has a list of things that should be included in a protocol for a trial design, but the first statement in this section says the scientific integrity of the trial, the credibility of the data from the trial, depend substantially on the trial design. So this is extremely important to get this right and to have it be comprehensive and inclusive. So I'm going to use the examples for the remaining portion of this lecture to talk about trial design. So we've talked about objectives. We're going to talk about primary and secondary endpoints, and that defines how the vaccine actually will be evaluated. The endpoints provide specific information on clinical or laboratory parameters that will be measured, and these endpoints are often further classified by intensity or severity of a reaction or seriousness of an outcome, or thresholds established for laboratory values that they will use in this measurement of endpoints. Trial treatment, and in this case, they mean the intervention, which is our vaccine. So you need to describe the trial treatment, and that means detailed description of the product, the number of vaccinations, when you're giving them, the dose, and the route in more detail than you would have given in just a little bit of the background. You're going to describe exactly what you're doing in this particular trial. And this text box shows a little snippet narrative from an experimental design for a novel influenza vaccine. So it provides, the name of the product, description of the product, and the dose. They're, in this case, giving 2 doses intramuscularly 3 weeks apart. Randomization is another important aspect of trial design. Basically, randomization is like flipping a coin to see how someone will be assigned to one group or another. And there are various randomization designs, and this particular one on this slide is a very simple one. There's a choice between vaccine or placebo, and no matter what you're assigned, you will get the product at the same time period. So you're enrolling 36 children, 30 will be randomly assigned to get the investigational vaccine, and 6 will get the placebo. Both products, vaccine and placebo, would be described in detail in the narrative description of the trial design. So this is another study design to evaluate the safety and immune response of a vaccine given at different schedules. So there are 3 groups with products given at different time points. In Group A, they will receive product at 0, 3, and 6 months, Group B at 0, 1, and 3 months, and Group C at 0, 1, and 6 months. But you can see if you look at the table, within each group, volunteers are also randomly assigned to get vaccine or placebo. This is a pretty common design. And this last one I'm going to show you is a dose escalation design that also can be found in vaccine trials. And in this one, they're giving a 25 microgram dose to 12 people initially. 10 would receive vaccine and 2 placebo. And they would do that study and then evaluate the results and then move on to higher doses to 50 micrograms or 100 micrograms in this particular trial. And the process of actually how you go about assigning those different groups to different volunteers will be described in the protocol in great detail as well. So who gets what? Let's talk about treatment assignment and blinding. So what is blinding? Well, GCP defines blinding as the condition when one or more parties are kept unaware of the treatment assignment. The reason they do this is to reduce bias in a clinical study, and the blinding can be done in a few ways. We can have single-blinded, where the volunteer or observer is unaware of the assignment, and that can be for situations where the evaluators would not have an impact on biasing the results. Maybe it's just a lab test that you're looking at and not any other kind of responses. You can have an open label trial where everyone knows what you're getting, and it may be in later phases of trials when you're perhaps evaluating when you give the product and everyone's getting the same thing. But the classical definition of a randomized controlled trial is a double-blinded study, and most vaccine trials in phase 1 through 3 are conducted in this double-blinded manner. So GCP defines double-blinded as the volunteer, the investigator, the monitor, and in some cases data analysts are unaware of the treatment assignment. That sounds like a lot more than double, but it refers to the fact that both sides, the volunteer side and the individuals who evaluate the response, are blind to the treatment assignments. But someone knows the code. So it's blinded to those that are doing the evaluation, but a code does exist. So a protocol would also include details on how you would maintain the blind. So whether a product is coming in a blinded way, the vials may be coded, pre-labeled. There may be someone who is an unblinded vaccinator if the products look too dissimilar. But this would be described in the protocol, as well as a description of any unblinding procedures. Protocols always have unblinding procedures. It will describe who has the randomization codes, whether it's a coordinating center, and when, in what circumstances, you may break the blind and unblind the investigators and or participants in the trial. So another huge portion, I'm going to end out this section talking about trial procedures. And this is a very large portion of a protocol, and we're not going to spend time talking about every little procedure, but just to emphasize that this would be very detailed, describing every step along the way. So from the screening process through the study visits, the clinical evaluations that you do, the specimen collection, the laboratory evaluation, how you process specimens, store them, ship them, all of that includes methods, timing, supplies, and very detailed descriptions of how you will conduct this. Some of these details may be found in a study operation manual, but the consistency of the protocol, we talked about that, trial design and consistency of the protocol, depends on the clarity of this information. So it's very important. But it's an awful lot to process, so I always like to look at the schedule of procedures, and most protocols have something like this. On this slide I have a table, the schedule of procedures or schedule of visits it may be called, and it's a common appendix, and it has an overall map of the study visits and the procedures that will be followed. So, it has all the procedures, and then across the top it has all the different time points, and when you actually do each procedure is indicated by an X, so that you're able to know, okay, this is when someone's coming in on week 4, this is exactly what happens. So this actually is a great place to start when you pick up a protocol, to take a look at the protocol schedule of procedures to see really a framework for the trial. So that's going to conclude Section A of this vaccine protocol lecture. In the next section, we're going to pick up with selection of subjects.